Hello everyone. Welcome to the second lecture of immunology. In this lecture, we are going to study the cells of the immune system. These cells are the real soldiers which are present in each and every tissue, and they are the real one who are giving you protection against any pathogen. What we are going to study today is about their characteristics and functions and how they are important. Okay, so please don't fast forward this video, otherwise you may miss some crucial information. Let us start the lecture, and before going forward, let us see what we studied in our previous class. So let's have a quick recap. So what we started in the class was about the discussion on immune system and how immune system is important, what is immunity, what is immunology, and how it came into the existence through some history. Then we studied the humoral versus the cellular immunity, their features, and how these two are interrelated. We also studied few important concepts of mammalian immunity, including the clonal selection. We also studied the two branches, the innate and the adaptive immunity, and how innate immunity is giving us the first line of defense, as well as it is also communicating to the adaptive branch about the infection, and it is alerting and preparing the adaptive branch. We studied the primary and the secondary response and how these are important also in terms of the vaccination. And then we saw some unusual immune responses like immune dysfunctions. Okay, so now we are moving ahead, and now we will be discussing the different cells. So we will go deep into the details so that slowly we will start learning about the core concepts in immunology. So let's first start with the organization of our immune system. If we see the immune cells, they are the most dynamic cells in the body. Okay, they are the most dynamic cells in the body, and they monitor each and every tissue, wherever, whatever inside your body. They monitor each and every tissue and cell, and they organize the immune response. If they find any pathogen or if they find any irregularity in the tissue, they will definitely respond. They arise from stem cells. All the immune cells, they arise from stem cells, and stem cells are the mother cells of the body. They are the cells which are actually defined by two characteristics. So they have two characteristics which make them any cell a stem cell. What are these two characteristics? The first is the self renewal. They can make their undifferentiated copies, so they can self renew. Now when we are saying they can make their undifferentiated copies, that means they can differentiate into all diverse cell types, whatever cell types we have inside our body. Stem cells can differentiate into them. So they are the undifferentiated cells which can self renew, as well as which can differentiate into all diverse cells inside our body. These two characteristics make any cell a stem cell. Fine. Now, if we see, a stem cell can divide into two daughter cells which have the same characteristics as the mother cell. So one stem cell can divide into two stem cells. This process is known as self renewal because it is making two copies out of it. And these two copies can again either differentiate or they can self renew. The second sort of division can be where the stem cell is dividing into one stem cell, which is the one daughter stem cell, and another is the committed cell or the progenitor cell. And this committed cell, it can form any terminally differentiated cell depending on what kind of microenvironment it is getting. And the third kind of division where one stem cell can give rise to two committed cells. So here it is not self-renewing, rather it is committing itself so that the daughter cells can form different cells which are more differentiated, right? So if you can see, when one mother cell is giving rise to two cells which are of same kind, so this is also stem cell, this is also stem cell, or here you can see this is the committed cell and this is also the committed cell. We call this kind of cell division as a symmetric cell division. Okay, so this kind of cell division is symmetric cell division, and where the mother cell is giving rise to two different daughter cells. Here in this case it is one is the stem cell but other is the committed cell. We call this kind of cell division as asymmetric cell division. This is the asymmetric cell division. So stem cells can either divide symmetrically or they can divide asymmetrically. Clear? Okay. Now, if we see our immune system, then we have the lymphoid organs, and these lymphoid organs are the organs where our immune cells develop. So these are the primary lymphoid organs where our immune cells develop, and these the two examples are the bone marrow and the thymus. Okay, so lymphoid organs are those organs where our immune cells develop, and they are also the site of the immune response. And the secondary lymphoid organs are the sites where the response is initiated, and the examples are spleen, lymph node, gut associated lymphoid tissue, and mucosa associated lymphoid tissue. Actually, mucosa associated lymphoid tissue is the general term under which this GALT comes, or there are some different other kinds of associated tissues, what we will see in today's presentation. Okay, so in this slide what you have seen is that immune cells are the most dynamic cells, and they all arise from the stem cells, and then stem cells have two properties, which is the self renewal and differentiation. And then there are two kind of lymphoid organs, one are the primary lymphoid organs and other are the secondary lymphoid organs. Now, if we see the hematopoietic stem cells, these are actually the stem cells which give rise to a number of different cells. These are the well studied and the model stem cells. Okay, so these were the earliest known stem cells which were identified. They are pluripotent and they make all sort of cells which are found inside the blood. So all the blood cells, they arise from the hematopoietic stem cells, and they maintain rarity. That means they are very rare, and if you can see the rarity, they are less than 20 cells per million cells in bone marrow. So that means if you start counting the bone marrow cells, and if you count around a million cells, that is equivalent to 10 lakhs, then you will find around 20 cells or less than 20 cells which are of the hematopoietic stem cells categories. And so they are very, very rare, right? But they are very, very potent. Now, normally they are quiescent. Quiescent means dormant. They show either no or very few cell division. They remain in the G naught state, but they are metabolically active. Although the activity is low, but they are metabolically active. And very few cells divide, okay, because they have to maintain their rarity and they have to self renew. But during infection, whenever there is an infection, they show enormous proliferation. They divide rapidly. And they divide rapidly because our body requires certain cells. And those cells arise from these stem cells to fight the infection. So, during infection, they show enormous proliferation. Now, they are lin negative cells. So, lin is the lineage specific markers, and these lineage specific markers are absent. Whenever a marker is absent, we say negative. So here you can see this is the negative sign, and this lin is the marker. This is called as lin negative. and that means the lineage specific markers are absent, and these lin markers, they are employed for the isolation. Actually, they were very useful when we first isolated the hematopoietic stem cells. How? I will show you. Now if you see here this picture, here, what scientists did, they took the plastic plate, maybe a 24 well plate or 48 well plates, and then they coat the plate with lineage specific antibodies. So all the antibodies here you can see here, they all are lineage specific antibodies. That means some will bind to some cells, others will bind to other cells which are fully differentiated. Okay, so now here you can see the different cells, like N stands for neutrophils, E stands for eosinophils, but this sky blue color cell, it is S, and this is the stem cell. So this is the stem cell, and then you can see these gray color cells which are P. These are the progenitor cells. Okay, they are the progenitor cells. What are these cells? You don't need to worry, we will discuss in the later slides, but you remember that this sky blue color cell is the stem cell, and these gray colored cells are the progenitor cells, and they are found in bone marrow. BM is the bone marrow. So bone marrow contains a mix of cells, and these mix of cells, we plate on a, a pre-coated plate which is a plate which is pre-coated with the antibodies, and these antibodies are lineage specific. Now what will happen? We will culture for 1 or 2 hours. All the cells which have the antibodies here, they will catch and they will bind. So you can see here that we have eosinophils, right? Then we have neutrophils, then we have lymphocytes, then we have the B cells, then we have the macrophages. Okay, these cells have already been bound to the antibodies. But some cells, like the progenitors and the stem cells, they are free. So what we will do, we will discard what binds here. So we have no interest in these cells because they are already terminally differentiated cells. But we collect the cells which are the unbound cells. And when we stain and observe under microscope, we will find that these cells are the lineage negative cells, and they are the mix of stem cells and the progenitor cells, and that's how we discovered and isolated these cells, right? So if we see the hematopoietic stem cell markers, they are the lin negative, that means lineage negative, then SCA1 positive. Now this is a protein SCA1. It is a positive. Hematopoietic stem cells, they express this marker, and that's why it is a positive. So all the markers are positive which are expressed on a cell. All the markers are negative which are not expressed or which are absent on a cell surface. So lin is a marker which is absent, so we have written lin negative. SCA1 is a marker which is present, so we have written SCA1 positive, and then the third is the c-kit which is also present, so it is a c-kit. Together we call the phenotype of the cell is the LSK phenotype. L stands for lin, S stands for SCA1, and K stands for c-kit. Okay, plus these cells, the hematopoietic stem cells, they also show CD34, expression of CD34 positive. So that means this particular protein is also present. So if together we see the characteristic of stem cell, particularly the hematopoietic stem cells, they are lin negative, SCA1 positive, c-kit positive. That means they show LSK phenotype, plus they are CD34 positive. Okay. Now if you see the c-kit, it is a cytokine SCF receptor which promotes the multipotent progenitor, and SCA1 is a stem cell antigen. Okay, so now we have different type of hematopoietic stem cells. One is the long term hematopoietic stem cells. Long term. We call it as a LT. It is the most quiescent and it remains pluripotent lifelong. These are the characteristics of these long term hematopoietic stem cells. Most quiescent and lifelong pluripotent. Then they are converted into short-term hematopoietic stem cells. And these short-term hematopoietic stem cells are less quiescent and they show frequent divisions and they have very limited self-renewal capacity. So they are now moving or progressing towards some specificity. Okay, but still they are the hematopoietic stem cells, but they are for short term because they are going to convert into some progenitor cells. So long term hematopoietic stem cells, they maintain the pluripotency and self renewal in the pool. Okay, but short term hematopoietic stem cells, they are less quiescent and they show frequent divisions, right? Now these short-term hematopoietic stem cells, they give rise to the multipotent progenitors. They give rise to multipotent progenitors. And these multipotent progenitors are very limited self-renewal capacity. They have a very limited self-renewal capacity. And they show rapid proliferation. And they can differentiate into either lymphoid or myeloid lineage. Okay, so multipotent progenitors, they are the result of the short-term hematopoietic stem cells, which are the result of the long-term hematopoietic stem cells. Fine. So by the now you by this time you should be knowing that we have the hematopoietic stem cells which give rise to all the blood cells, and we have long term and short term hematopoietic stem cells based on their quiescence and the pluripotency they show, and of course the cell division. And then we have the progenitor cells which can progressively differentiate into either the lymphoid lineage, the cells of the lymphoid lineage, or the cells of the myeloid lineage. Okay, now what are the cells of the lymphoid and myeloid lineage? Now you will come to know. So this is the hematopoietic site in mouse and in embryo. So you can see both the embryos, they are almost at the same stage. You can see here the placenta, right? There, here it is the yolk sac, which is the yellow color. Here you can see the fetal liver, which is of this pink color. And then you see here is the AGM. Okay, what is AGM? I will just define it for you. Now, look at the picture. So hematopoiesis, which is the formation of the blood cells. Hematopoiesis begins in the yolk sac. So here you can find the yolk sac, which is the yellow part. This is the yolk sac, and here it is the yolk sac. Okay, here. So it begins in the yolk sac 7 days after fertilization in the mouse and 3 weeks after fertilization in case of humans. So after 7 days and 3 weeks in mouse and humans, in the embryo, the blood cells start forming. Okay. Next, these fetal hematopoietic stem cells, they are also present close to the developing kidneys, and this region is known as aorta gonad mesonephros region. So it is known as AGM. The full form of AGM is aorta gonad mesonephros region. And when you can find this particularly when the fetal heart rate starts beating, because now the heart is formed and it has started beating, so that means it has started pushing the cells to the tissues so that it can develop the circulatory system, right? So when the heart starts beating, we can find these hematopoietic stem cells near the kidneys, developing kidneys in the AGM region. Now, in the later stages of the fetal development, it is the placenta And finally, at the maturation of the embryonic development, it is the fetal liver where you will find the hematopoietic activity. So here you can see initially it started in the yolk sac, then AGM, then placenta, and finally it is the fetal liver which is taking over here, and now here exclusively in the later stages till birth, that the fetal liver is the major site of the hematopoiesis. And after birth, it is the bone marrow where the hematopoietic activity takes place lifelong. Right? So these are the different sites of the hematopoiesis in case of mouse and in humans. Now, if we understand the potencies of the stem cells, because there are different potency levels, so we will come with the term totipotency. Now, totipotency you must have heard, particularly it is used in the plant cells, but here in animals we have the zygote, which is a totipotent cell, and totipotency is the highest differentiation potential. That means any cell which can give rise to the entire organism, they are the totipotent cells. And zygote is the example because zygote is the single cell which is formed after the fertilization, and this cell gives rise to the entire complete organization of a animal. So these totipotent cells, they can develop into any cell type in an organism, including, now you have to remember, including extra embryonic tissues, for example the placenta, or a complete organism. So any cell which can develop into a complete organism, or it can also give rise to extra embryonic tissues, that cell is called a totipotent cell. Then comes the pluripotency. Now, pluripotency, they can develop into any cell type in an organism, but not extra embryonic tissues. So, here the important part is that extra embryonic tissues are not formed by the pluripotent cells, although it can also develop into any cell type inside the body. Then comes the multipotency, and they are more specialized cells which show limited differentiation potential, and they can give rise to the cells of a very narrow lineage. Okay, so if you see here, this is the zygote which can give rise to uh, embryonic cell, and the earlier embryonic cells are the pluripotent cells like embryonic stem cells. These embryonic stem cells, they can give rise to the three germinal layers: ectoderm, endoderm and mesoderm. So that means they can develop into almost any kind of cell. And then in multipotency, the cells of the ectoderm will give rise to the different terminally differentiated cells of the ectoderm, and the endoderm will form the cells of the different cells of the endoderm, and the mesoderm can form the different cells of the mesoderm, right? And in case of the unipotency, it is the ability to differentiate into only one specific type. So here you can see the one single terminally differentiated cells. The precursors of the terminally differentiated cells are the unipotent cells. Okay, remember, the precursor of the terminally differentiated cell is a unipotent cell because it gives rise to only one single kind of cell, and that is the terminally differentiated cell. So here you can see the neural stem cell is giving rise to the neurons, although through a series of intermediate progenitors. And here you can see the gut cell, which is the terminally differentiated cell, and the RBCs, they are also the terminally differentiated cells. Now, if we see the hematopoietic stem cell differentiation pathway, what we see here, that we have the hematopoietic stem cell, and this hematopoietic stem cell can self renew itself. Okay, because they show two characteristics: one is the self renewal and another is the differentiation. So they can self renew, and they can further differentiate into either the myeloid progenitor cell or the lymphoid progenitor cell. Okay, so it can form either myeloid or lymphoid progenitor cell, and these myeloid progenitor cells can give rise to a number of progressive progenitor cells, for example, granulocyte monocyte progenitor cell, which can give rise to further either neutrophil or monocyte or dendritic cell. Okay, so one important part which you should remember here is that this sky blue region in the picture, it is the bone marrow. So whatever is happening here, the hematopoietic stem cells, when it is giving rise to the myeloid progenitor cell, or it is giving rise to the different further progenitor cells, every differentiation is happening inside the bone marrow. But when they are released in the peripheral tissue, they form the differentiated or terminally differentiated cells which are found in the peripheral tissue. Okay, so here the granulocyte monocyte progenitor, it is forming the monocytes and releasing into the blood, and then this monocyte is going into the tissue forming macrophages, and it can also form dendritic cell, or dendritic cell of the myeloid progeny can also be formed directly from the myeloid progenitor cell. Now, eosinophil progenitor can give rise to eosinophil. Basophil progenitor can give rise to basophil. Then myeloid progenitor gives rise to mast cell directly. Megakaryocytes give rise to platelets, and erythroid progenitor gives rise to erythrocyte or the RBC. So this lineage is known as the myeloid progenitor, and these all cells are the myeloid cells. Now see the other side, which is the lymphoid progenitor, and these lymphoid progenitors can give rise to the dendritic cells. So these dendritic cells can also be formed not only from the myeloid progenitor but also from the lymphoid progenitor, and these are very important cells which we will see shortly. Then lymphoid progenitor cells, they can form B cell progenitor, and this B cell progenitor in the bone marrow, it can give rise to the B cells, and they are the B lymphocytes. Okay, and some cells migrate to the T cells, sorry, in the thymus they are called the T cell progenitors, and in the thymus the T cell progenitors give rise to the T cells, and we have the two subsets of the T cells which are very important. One is the T helper cells and another is the T cytotoxic cells. Okay, and these lymphoid progenitor can also give rise to innate lymphoid cells. So innate lymphoid cells are the different cells which are actually the lymphoid cells, but which can show the innate response, and one of the examples is the NK cells or the natural killer cells. This is one of the examples of the innate lymphoid cells. So we will also see these cells. Okay, so now you have understood that we have the hematopoietic stem cells which can either give rise to myeloid progenitor or lymphoid progenitor, and from the myeloid progenitor we can give rise to these cells can give rise to the macrophages, monocytes, dendritic cells, neutrophils, basophils, okay, eosinophils, mast cells, platelets, erythrocytes, and on the other side the lymphoid progenitor can give rise to dendritic cells, T helper cells, T cytotoxic cells, B cells, and a number of innate lymphoid cells. So myeloid cells are the members of the innate immunity. You should understand that, and you should remember that all the here cells you can find here, the myeloid cells, they are the members of the innate immunity. That means they take part in the innate response, which is the first line of response and which is non-specific. I am sure you must remember this thing. And the B and T cells, they are the members of the adaptive immunity. Okay, so these are the members of the adaptive immunity, very specific responses, and they are basically primed, or they are induced by the innate cells. Then we have the ILCs, and these ILCs, they are the innate plus adaptive immunity features. So these cells, as I told you, for example, I give you an example of the NK cells. They not only show the adaptive immunity features, but they also have some innate features. So they are the different categories. Okay. Megakaryocytes and erythroid progenitors, they can arise directly from the long-term and the short-term hematopoietic stem cells. Okay, they can arise directly from the long term or the short term hematopoietic stem cells. And the differentiating cells, they undergo lineage commitment. Now this is a term, lineage commitment, which you will be hearing frequently in our subsequent discussions. Lineage commitment means where a series of irreversible changes that progressively restrict their potential. So that means here hematopoietic stem cell, it has the potential to differentiate into any kind of cell. It can differentiate into dendritic cell, it can differentiate into monocyte, it can differentiate into neutrophil, or it can differentiate into the T helper cell or the T cytotoxic cell. It can differentiate into any cell. But as it is dividing and more committing, then it is restricting its ability to convert. For example, if here the hematopoietic stem cell becomes myeloid progenitor, then now myeloid progenitor cannot give rise to the T cells or the B cells or the ILC. It can only give rise to the different cells of the myeloid progeny. And if the hematopoietic stem cells, it differentiates into lymphoid progenitor, then this lymphoid progenitor can give rise to the cells of the lymphoid progeny. That means the dendritic cells, the lymphoid dendritic cells, T cells, T cytotoxic cells, B cells, or the innate lymphoid cell. But it will not give rise to any of the progenies from this myeloid cells. Okay, so it has restricted its ability to differentiate. This is known as lineage commitment. Now it is more lineage specific cell. And further, these cells, the T cell progenitor, it can either give rise to the T helper cell or T cytotoxic cell, but not the B cell or the innate lymphoid cell. So that means it has further restricted its lineage commitment. Now it is more committed towards making only T cells. So this is known as the lineage commitment, where by progressive cell division and differentiation, the pluripotent stem cell, it slowly becomes progenitor and then finally becomes the terminally differentiated cell. Okay, I hope you understand these intricacies in a very easy manner. Okay, but remember that, because that will be keep coming. Now we will see how these hematopoietic stem cells are differentiated and how the differentiation is regulated. So you can see here we have the long term hematopoietic stem cell, then we have the short term hematopoietic stem cell. So this long term hematopoietic stem cell gives rise to short term hematopoietic stem cells, and short term hematopoietic stem cells give rise to multipotent progenitor cell. Now, these multipotent progenitor will give rise to either myeloid progenitor or lymphoid progenitor, and then they can give rise to further terminally differentiated cells of their respective progeny, either the myeloid or the lymphoid one. Okay, in the last class in the last slide we just saw this. Now, how they are regulated? They are regulated by certain transcription factors. So hematopoietic stem cell genes have easy accessibility to the transcription factors, and the environmental signals determine the right set of transcription factors to be upregulated. That means what kind of transcription factors will be formed or will be upregulated or will come up into the picture, that will be decided by the environmental signals. For example, if there is a case of anemia, in case of anemia, the number of the RBCs will go down. And when the number will go down, then the cell will demand more RBCs because they have to carry out the function of the gaseous exchange, which is the primary function. So the cells, and the environment and the body, it will start releasing certain factors, some cytokines, which will actually drive the hematopoietic stem cells to form RBCs. So the environmental signals will determine the right set of transcription factors. So those transcription factors which will drive the synthesis of the RBCs, they will be upregulated. Okay, and others will be downregulated. and once they will be upregulated, they will bind to the respective genes, and then they will start transcribing those genes. Okay, so the transcription factors are the major drivers of the regulatory pathways. Okay. Now, these transcription factors, they regulate whether the cells will remain in the quiescent state, or whether it will proliferate, or whether it will differentiate. Okay. So differentiation, proliferation, and quiescence, everything is regulated by the transcription factors. Now, in order to know what are the transcription factors, scientists try to purify these transcription factors, and they found that the top 10 transcription factors are the GATA2, the RUNX1, the SCLTEL1, LIL1, LMO2, MEIS1, PU.1 or PU.1, ERG, FLI1, and GFI1B. Okay, so these are the transcription factors which are responsible for the major regulatory pathways, and which regulate the quiescence, the proliferation, and the differentiation of the hematopoietic stem cells. Now we will see some examples here, like these Icros and GATA3. They are responsible for the lymphoid progenitor. Okay, while uh, PU.1 and GATA1 towards the myeloid progenitor. Now here you can see the granulocyte monocyte progenitor. These two transcription factors are important, and here for erythroid progenitor this GATA1 is important. For T cell progenitor it is the Notch1 and GATA1, and then for the B cell progenitor it is PU.1, PAX5, and EBF. Okay. Now some details here. Icros, for example, this Icros is required for lymphoid development, but not for the myeloid development. Okay, now remember, it is required for the lymphoid development, but not for the myeloid development. And in the absence of Icros, animals are immunocompromised and they cannot mount a full response. So how important this Icros is, you can see, right? Then PU.1, the low levels cause lymphoid development and the high level cause myeloid development. So it is not only the absence or the presence of any transcription factor, but their concentration also drives the cells to a specific pathway. Now here is an example. The low levels of PU.1, it is causing the cells to drive towards the lymphoid development, and the high levels are causing towards the myeloid development. So it regulates the choice between erythroid and the other myeloid lineages. So it's a very important transcription factor, PU.1. Now, if you see the Notch1, Notch1 drives the T cell development. It is important for the T cell development. And the GATA1 drives the multipotent progenitors toward the erythroid development. And it forms the RBCs further. Okay, so it forms RBCs, right? So here you saw, some regulatory aspect. Now, if we see the release of mature cells from the bone marrow is responsive to circadian cycles, which are the day and night cycles, and they are also regulated by the sympathetic nervous system. Okay, now this is really important because our mature cells, when they are released into the system after the terminal differentiation, that is also regulated not only by the transcription factors but also by the circadian cycle. Now look at the picture here. Here around 6 a.m. there is a onset of the light. There is a dawn. Okay, and when at this time noradrenaline and TNF it bursts, and in its presence the hematopoietic stem and progenitor cells, HSPC is hematopoietic stem and progenitor cells, they differentiate and they form the terminally differentiated cells, and then these cells are released into the blood. They are released into the blood. Fine. Now in the night there is the increased concentration of the melatonin in the bone marrow, And this increased concentration of melatonin in the bone marrow causes the hematopoietic stem cell to self renew. Okay, so here in the daytime, while our hematopoietic stem cells they were busy forming different cells, in the night under the melatonin concentration they were self renewing, so that they maintain the rarity and they maintain their presence in the pool. So it is not only the transcription factors but also the circadian cycle which is regulating the release of the mature cells and the self renewal. So here that is a important aspect. Then the environmental changes also impact the rate of hematopoiesis as well as the production and release of the specific cell lineages. Now, when there is an infection, it can result in the release of the cytokines that enhance the development of the myeloid cells, including the neutrophils. So when if there is an infection, we need neutrophils. Okay, neutrophils are there. There will be the upregulation, and if there is, as I told, in case of anemia, we need RBCs. So RBCs will be there. So depending on what kind of demand is there inside our body, our cells they make those particular cells from the stem cells, and that is regulated, and that is maintained by the environmental signals. Okay, now we will see the different blood cells. By now we understood some fundamentals about the blood cells and how they are formed, and our discussion was more focused towards the stem cells. Now we will see the different blood cells. Now, if you see the traditional identification method, we use dyes, and we use hematoxylin and eosin. So we call them as H and E stains. And these stains are still very common and useful in the lab. If we want to do some primary or some very basic analysis, we can use these two dyes, hematoxylin and eosin, and we can find the different cells on a slide. Okay, now these two dyes they highlight the intracellular differences because their pH sensitivity and different affinities for charged macromolecules in a cell, because these dyes they act differently in different pH, and that's why they can highlight the differences in different cells. And when these differences are highlighted, you can under the microscope easily recognize the phenotype of the specific cell. Now, hematoxylin is a basic dye. Okay, it is a basic dye, and it binds to the basophilic nucleic acids and stain them blue. So all the cells or all the macromolecules or all the cell parts or organelles which take up hematoxylin, they will become blue under the microscope. While the eosin, it is a kind of a acidic dye, right? And they bind to the eosinophilic proteins in granules and cytoplasm, and then they stain them pink. So you can see both these dyes they help by the color differentiation. One will show the blue color in a cell and another will show the pink color under the microscope. And why it is happening? Because hematoxylin is a basophilic dye while eosin is an acidic dye. Okay. Flow cytometry and immunostaining, these are the two advanced methods, and they have actually improved our accuracy of determining and deciding what kind of cells these are. And flow cytometry can pinpoint each and every cell based on the monoclonal antibodies. So here in this technique we use monoclonal antibodies which are linked with a fluorescent dye, and then these monoclonal antibodies they are incubated with the cell culture, and they bind to their respective receptors or antigens on the cells, and then under the laser light, okay, each cell like a file, a small file, in a single linear, one after another. Each cell, it moves, and then it is detected based on, the kind of monoclonal antibody it has bound. So we can easily determine, what kind of cells and how many cells are there in the culture. And then immunostaining is the method where we stain the cells using the monoclonal antibodies, and we see this binding with the help of the fluorescent dye, maybe under the microscope, fluorescent microscope. So these two methods are the advanced methods, and they have improved our understanding and accuracy about the detection of the different blood cells in a culture or in a sample. Okay. Now you can see the different cell types and the numbers there. So RBCs and platelets, we don't have in terms of the total percentage because they are the unique cells. And but the leukocytes are the white blood cells, and we have different white blood cells like neutrophils, lymphocytes, monocytes, eosinophils, and basophils. What you can see here is that the neutrophils are the most abundant WBCs. They are about 50 to 70% of the total WBCs. So these neutrophils, they are the most abundant. Okay. While in case of basophils, you will find they are the rarest cells. Okay. They are the rarest cells. So these two things you should remember: neutrophils, the most abundant cells, and basophils are the rarest WBCs, which is they are found in the circulation. RBCs and WBCs. So RBCs are the erythroid cells. They are the cells from the common myeloid progenitor. Now you remember that the cells from the common myeloid progenitors, or we call them as a CMP, they give rise to RBCs, and they give rise to the leukocytes or the WBCs. So these WBCs or leukocytes or myeloid cells, they are the first responders to the pathogen. Whenever there is an infection, the first cells to reach at the site are the leukocytes of the myeloid progeny. Okay, and they communicate the presence to the cells of the lymphoid lineage. So that means, that we have the cells B cells and T cells which are of the lymphoid lineage. So these cells or leukocytes, they communicate the presence of the pathogen to the lymphoid cells, the B cells and the T cells. So these leukocytes of the myeloid origin, they are the innate cells. Okay, we call them as a innate cells because they take part in the innate immunity. Now we have four type of these leukocytes: granulocytes, monocytes, macrophages, and dendritic cells. These four type of cells are the innate cells, and they are the myeloid leukocytes because their origin is the uh, myeloid progenitor cells or the common myeloid progenitor cell. So if we see the granulocytes further, then granulocytes are also of four different kinds. They are the frontline attackers, or they are the first responders. And these four types are the neutrophils, basophils, mast cells, and eosinophils. Okay, these four cells, they come under the group called as a granulocytes. And why we call them as a granulocytes? Because if you see under the microscope, you will find a lot of granules inside them, right? And that's the reason they are known as the granulocytes. So they can be categorized, and they can be differentiated based on the kind of granules found inside them. And all these cells have the multi-lobed nuclei. So you can see here two lobes. One lobe is this and one lobe is this. So you can see that in this particular picture, the nucleus has two lobes. So they have the multi-lobe nuclei, and they are very visually distinctive and distinguishable from lymphocytes whose nuclei are round. So lymphocytes are the small cells and they have the round nucleus, whether they are the B cells or they are the T cells. These lymphocytes, they have the almost same phenotype. So under the microscope, it is very difficult to determine which cells are the T cells and which cells are the B cells because they have the similar phenotype. But in case of the granulocytes, it is easier. Why? Because they have the multi-lobe nucleus, so we can easily define. Moreover, they have the granules, and granules have the different patterns, specific patterns, of these cells. What we will see now. Now let us start, by some more details. These are differ by staining characteristics because their cytoplasmic granules are different and therefore their staining characteristics are also different, and as well as the nucleus which contain, you know uh, these cytoplasmic granules, they contain the proteins, and these proteins have distinct functions. So now you can see here it is the neutrophil. So you can see the multi-lobed nucleus. So you can see here the dark part. This is the actual neutrophil under the microscope, and this is blue. Okay, so this is the multi-lobed nucleus, and then you can see the pinkish granules here. Okay, small granules here. Fine. So here, this is a representative picture where you have the multi-lobed nucleus, you have the granules, and then phagosomes. It shows that these neutrophils are the phagocytic cells. Then you have the basophils, and here in the basophils you will find that the nucleus has this different lobe shaped, and then it is filled with these dark stained granules, right? In case of eosinophils, you will find the granules and the pattern. Okay, and you can see here it has a bilobed nucleus here, and these are the pink stained larger granules. So if you can find the difference, you will see in the neutrophils the granules are lightly stained and they are smaller as compared to the granules which are darker. Okay, in case of eosinophils. And in the basophils, the granules are too much and they are obscuring the nucleus. So you can see here the two lobes. I can see one lobe is like this and another lobe is like this, but all around there are the small granules which are darkly stained. Now, if you see the mast cells, so mast cells, the nucleus is surrounded by a very dark stained granules. So, if you notice the differences, you should see the shape of the nucleus and then the number, color, and shape of the cytoplasmic granules. And once you will see here, you will find the difference. And these differences, they are easily, very distinguishable features under the microscope, right? Now let us focus on each and every cell here. So let us start with the neutrophils. So neutrophils are the most abundant cells, and around 50 to 70% of the WBCs are the neutrophils, and they are also known as the polymorphonuclear leukocytes. So we also call them as a PMNs, right? They are formed in the bone marrow, then they go to the peripheral blood, and in the peripheral blood, they stay around 7 to 10 hours, and then they move to the tissue and they remain in the tissue for few days. So that's how they start from the bone marrow and then they finally they reside in the tissues. There are 3 to 5 lobed nucleus. So you can see here the nucleus. This is the lobes of the nucleus. So you can see the 5 lobes here. Okay. And this is the 5th lobe here. And they phagocytose bacteria. So they are phagocytic cells. Okay. Chemokines complement during innate response increase numbers because when there is an infection, a number of cytokines and complements, they are the part of the innate immunity. They increase the number, and this is known as neutrophilia. When the neutrophil numbers are increased inside the blood, the situation is called neutrophilia, and that indicates the infection. So when in a blood profile there is an increase in neutrophil, doctor can see that there is a bacterial infection inside the body. Sometimes the low count can also be seen, and this low count is known as neutropenia. Okay, this is known as neutropenia, and it shows the severe infection. If there is an infection, then the infection is very severe. And why the numbers are low? Because all the neutrophils and most of the neutrophils, they are fighting with the infection, and that's why the numbers are very low. They are either dead in the battle or they are still fighting, so the numbers are very low. And if there is no infection, there are no symptoms of infection, then there is some problem with the bone marrow, so there is a clinical issue. So doctors can easily decide whether it is a clinical issue or it is a severe infection based on the symptoms. Okay, and low count is known as neutropenia and high count is known as neutrophilia. They are the main cellular component of pus. They are the main cellular component of pus, and pus is a thick mix of debris, cells, and microbes. Pus is a thick mix of debris, because whenever there is an infection, fighting goes on, and when the fighting is happening, then definitely there are the dead cells, and there are some debris also, and then there are the microbes and the cells. Both are, they are there, and plus debris is there, and they form a type of mix which is a pale yellow, and under the skin usually you can find the pus formation, and that indicates that this is the site of the infection. If you see the granular content, because these are the granulocytes, so they have the antimicrobial proteins, they have the proteases which are helpful in tissue remodeling, they have protease inhibitor to control the proteases. Okay, and they have the histamine, and these histamines, they have the function of the vasodilation and inflammation. So these granular content, they are immediately released whenever they hit any bacteria or they find some right signals, and but the mix of the content, it causes the degradation of the infection or the bacteria. They play a role in adaptive immune response because they are the innate cells, and innate cells, they communicate to the adaptive cells so that they can also come into the picture and they can start fighting with the infection. Now we will move to the basophils. So basophils are the rarest cells, okay, and you will find just around 5 in number per cubic millimeter of the blood. But they are very potent responders. Despite of their low number, they are very potent responders. They are non-phagocytic cells. They have large granules. You can see the large granules here. Okay, they are filled with the granular content, and their nucleus is 2 to 3 lobed, right? So you have 2 to 3 lobed nucleus is there, and they are structurally and functionally similar to mast cells, but their lineage is different. Okay, earlier it was a thought that the mast cells and these basophils, they belong to the same lineage, but later it was found that they although are structurally and functionally similar, but they are different. Lineage is different. They release granular content in response to binding of a circulating antibodies. Okay, and they have histamine, which causes vasodilation and inflammation. They have heparin in their granules, which is an anticoagulant, and they have leukotrienes, which regulate the inflammation. So histamine, heparin, leukotrienes, they are the important granular content. They play a role in controlling parasites, and particularly the helminths. They have an active role in controlling the parasites. So whenever there is a parasitic infection, you can easily assume that basophils are on action. They stimulate IgE secretion in some cases. So IgE causes the inflammation and allergy. Okay, so they stimulate the IgE secretion. They express IgE receptors, and when the IgE is there, okay, they bind the IgE and then cause the release, and the release can kill a kind of infection, usually in the parasitic infections particularly. And when there is some sort of inflammation, then Ig can rise and they can cause the release of the granular content from the basophils. They also secrete IL-4, IL-13, which modulate adaptive immune response. Okay, so these two important interleukins, 4 and 13, they modulate the adaptive immune response. Now you see the mast cells, and mast cells, the most of the granules you can see, they surround the nucleus here, and they are released undifferentiated from the bone marrow. So important part is they are released undifferentiated from the bone marrow, and they mature only after leaving the blood for the tissue. They have a large granules, and their nucleus is obscured by granules. So even here in this picture you can find the nucleus clearly, but if you see under the microscope you will find that even these granules are here, and they are obscuring the nucleus because the granular content is very high in mast cells. There are two types of mast cells. One is the MCT cells, where the granules are filled with mostly tryptase. So we call them as a mast cells tryptase, MCT cells. And then we have the mast cells tryptase as well as chymase and carboxypeptidase. So we call them as a MCTC cells. Mast cells tryptase, chymase and carboxypeptidase. So these are the two subtypes of the mast cells. If you see the granular content, they have the histamine, which is the major content, which causes vasodilation and inflammation, and heparin, which is an anticoagulant. Okay, leukotrienes, they regulate the inflammation, and then they have the tryptase, chymase, TNF alpha, which is the tissue necrosis factor alpha, and some other minor releases. They have the major role in inflammatory cascade. Okay, mast cells, they actually cause the inflammation, right? And whenever there is an inflammation, there will be the mast cells, and the content released by cross-linking with IgE antigen complex. Wherever there is an inflammatory response, IgE will be produced, particularly in case of hypersensitivity or allergy. And when they bind with the antigen, they form the IgE antigen complex. Once this IgE antigen complex is formed, then they cross-link, or the mast cells, they cross-link this complex and they release the content, and the major content is the histamine. This histamine, it induces WBCs, chemotaxis. That means the other leukocytes, they come at the site where there is a release of the histamine. Then airway smooth muscle constriction. Okay, so the airway smooth muscle constriction happens particularly when there is a bronchitis, and in case of bronchitis the feeling comes like somebody is strangulating your neck, your throat. It gives a very very discomfort feeling. Okay, that happens because of this histamine, and doctors prescribe antihistamines so that this feeling can be comforted, and it can be this activity is uh, shortened, or it is completely uh, eliminated. And then there is an increased vascular permeability. So histamine does wonders inside the body, and it is a mediator of the inflammatory response. Now we are coming to the eosinophils, and like neutrophils, they exhibit blood tissue migration. Okay, so they are released in blood and they can migrate into the tissue, and they are the most abundant in the small intestine. Remember, eosinophils are most abundant in small intestine. They are also the phagocytic cells, and they have the red and orange eosinophilic granules because eosins, they take up the eosin dye. So earlier they were called the eosins because they took up the eosin dye, and these granules, they are the eosinophilic granules. They take up the eosin dye. There are 2 to 3 lobe nucleus, and their most important role in the defense against multicellular parasitic organisms. All the complex parasites which come inside our body either through infection or through some other reasons, then these eosinophils, they take part in the battle with these parasites, including worms. They follow eosinophil chemotactic factor, which are released by the mast cells to reach the sites, because mast cells are already there and they have granulated not only the histamine but also certain factors, which is the eosinophil chemotactic factor, and they follow the path and they reach at the site. Their granular content contains eosinophil peroxidase, which is the EPO, which catalyzes the production of reactive oxidant to kill parasites. So this is a very potent, release. And then they have the major basic protein, a very unique protein which is in the granular content. These are toxic to helminths. Then they also have the antiviral ribonucleases. So that means they are capable of fighting few viruses. Okay. Then they also release the cytokines and chemokines for attracting other cells. Right. And they cluster around invading worms and damage their membranes by releasing the content. So their strategy is very clear. Whenever there is a parasite, these cells will cluster around the parasite and they will release the content. The content contains a number of important and very potent substrates like eosinophil peroxidase and the major basic proteins which are toxic, and they damage their membranes, and ultimately the degradation of the membranes causes the death of the worms. So they also secrete the cytokines to regulate the T and B lymphocytes. Okay, so this is the summary slide about eosinophils, and now let us move to the myeloid antigen presenting cells. Now, these myeloid antigen presenting cells are the antigen presenting cells of the myeloid progeny. So, these are a group of phagocytic cells from the myeloid progenitors, and these are monocytes, macrophages, and dendritic cells. And these cells are called professional antigen presenting cells or PAPCs. These professional antigen presenting cells, they form or they act as a bridge between the innate and the adaptive immunities. Right? Now, like all nucleated cells, they can present cellular peptides via MHC class 1 molecule, but unlike them, they can also present external antigenic peptides via MHC class 2 molecules. Now, you remember that all the nucleated cells in the body, they whenever there is some degradation, or there is some internal peptide to be presented to the outside cells, then we have the MHC molecules, which are the major histocompatibility complexes. So we have two variants, type 1 and type 2, right? So the type 1 variant is present in all the nucleated cells, and any internal, the cellular component, it is tagged with this MHC class 1 molecule, and then it is displayed on the surface. So that is done by all nucleated cells. But there are some cells which are special cells. So here the names are written, which are the monocytes, macrophages, and dendritic cells. They, besides the type 1, they also have type 2 MHC molecules. And these two type molecules, they can display the peptide of the external antigens. That means these cells can engulf the antigens, they can degrade it, and then they can present their component using MHC class 2 molecules. And these MHC class 2 molecules, they are used like a flag on the surface, and which are shown to the different lymphocytes so that they can act, okay, against these pathogens. So each APC can, or antigen presenting cell, because they can present the antigen, so we call them as an antigen presenting cell. So each APC can respond to pathogens, and it can secrete chemoattractants to activate other immune cells. So that is the major function of the antigen presenting cells. They are not only fighting themselves to the antigen, but they are also, giving a kind of signal using the chemoattractants so that other immune cells can come and get activated. Phagocytose pathogens and present antigenic peptides on their membrane surface via MHC class II molecules. And that's why they are called as the professional antigen presenting cells. T helper cells, they recognize antigen MHC2 complexes to initiate an adaptive response. So you just saw that we have the two subsets of T cells. One is the T helper cell and another is the T cytotoxic cells. So whenever these cells, okay, for example, suppose if I use here space, this is the cell, suppose this is the macrophage, okay, and this macrophage, it displays here a antigen, and this is MHC2, and this is the antigen. This is antigen. Now we have the T cells. This is the T helper cells. The T helper cells, they have the receptor to find out whether the MHC2 molecules with the antigen is there or not. The moment they come to know, they start secreting the cytokines. And these cytokines, this can activate the TC cells and the B cells. Okay, and that's how they do. So TH cells, they recognize antigen MHC class II complex to initiate adaptive response. Dendritic cells, they present antigen primarily to naive T cells. So naive T cells are those cells which are unexposed to any antigen yet. Macrophages and neutrophils, they efficiently remove pathogens and damaged host cells. So they also do the cleanup function. They are antigen presenting cells, but they are also, macrophages, they are also cleaning up the debris from the host cells. Now, if we see the monocytes, monocytes are a heterogeneous group of cells which migrate into tissue and differentiate into tissue resident phagocytic cells. Those phagocytic cells which reside in a particular tissue, they are called tissue resident phagocytic cells. They include macrophages and dendritic cells. So these tissue specific, tissue resident phagocytic cells, they are different from the normal phagocytic cells, that they also have some tissue specific function. Okay, so these are slightly different in terms of the functionality. So you can see here hematopoietic stem cells, they are giving rise to myeloid progenitor. This myeloid progenitor in the bone marrow, it is giving rise to directly the dendritic cells or the granulocyte monocyte progenitor. This granulocyte monocyte progenitor is giving rise to neutrophils or monocytes, and then these monocytes in the tissue, it forms the macrophages, or it can also form the dendritic cell. So that's how, the dendritic cells, monocytes and macrophages are generated by the granulocyte monocyte progenitor. There are two broad categories. One are the inflammatory monocytes, which enter tissues quickly in response to infection and differentiate into macrophage. For example, there is an infection in the tissue. So these monocytes, they get message by the chemotaxic. They immediately reach at the site in the tissue, and then they differentiate into macrophages and start doing the battle, okay, and cleanup. And then there are the patrolling monocytes, right? These are a smaller group that crawl slowly their main function is to patrol. Just like we have the patrolling police, they slowly move. You must have seen the police car or police bikes. They slowly move in a locality to just check whether the things are good or not. In the same manner, these are the patrolling monocytes that crawl slowly along the blood vessels, and they provide a reservoir for tissue resident monocytes in the absence of infection. So when there is no infection, they provide a reservoir for tissue resident monocytes. So monocytes which are present in the tissue, okay, and some function may be to suppress the immune response rather than initiate the immune response. So that is a question mark here. Still research is going on whether they are really, initiating the immune response or they are suppressing the immune response. But inflammatory monocytes are the monocytes class which immediately reach at the site of the infection and then start fighting with the pathogen. Now, the second cell is the macrophage. So macrophages, they are the differentiated form of monocytes. So monocyte in tissue can differentiate into specific tissue macrophages. And the example is like in case of bone, we have the osteoclasts. Osteoclast is the name of the bone macrophage. Microglial cell, it is the name of the central nervous system macrophage. And alveolar macrophage, it is in the lungs. So these are the tissue specific macrophages. So they play several different roles. They are the long term tissue residents and their main function is to repair and regeneration. These macrophages, they repair and regenerate the tissues and they are the long term tissue residents. Then we have the inflammatory macrophages. Their main function is to clear the pathogens from tissue and they act as a antigen presenting cells to activate the T lymphocytes, right? So macrophages, here you can see this yellow color, they are the bacteria or the tissue antigen. They are taken up slowly and here they are engulfed, and from here you can see with the lysosomal enzyme, this is the lysosome, they are degraded, and here you can see after degradation, they are degraded into small peptide fragments, and these small peptide fragments, they can attach with the MHC molecules, and these MHC molecules, they display some pathogen peptide here, and when it is displayed, that then the T helper cells, they reach and they recognize, and then they start secreting the cytokines, the same thing which we uh, shown in the previous slide. And these cytokines, they can either activate the T cells or the B cells. So activated inflammatory macrophages, they exhibit greater phagocytic activity, okay, as compared to the long-term tissue residents, whose main role is to repair and regeneration. They have the increased secretion of cytokines. They have the ability to activate T cells, and they are more effective antigen presenting cells for T helper cells, because our T helper cells, they need the right stimulatory molecules or the co-stimulatory molecules and the right set of signals so that they can help to induce the immune response. So they are more effective. Inflammatory macrophages are more effective. The receptors, they have the receptors for antibodies, and opsonized pathogens are readily ingested. So suppose there is a pathogen and this pathogen is opsonized, that means it is coated with the antibody, right? You can see here it is coated with the antibody. It can be taken up by the macrophage very readily, and it can increase the rate up to more than 4,000 folds. So you can see what is the magic these opsonization does in case of the macrophage engulfment. M2 macrophages, which is a population, they are mainly for the remodeling and the repair. They don't, take part in the battle. Rather, they remodel the tissue and repair the tissue, because somebody also needs that after the fighting, the tissue needs to be remodeled and tissue needs to be repaired and healed. So that is done by the M2 macrophages. So if you see, this is a list of the macrophages which might be, helpful for you to understand that, in tissues, what additional function they do. In lungs, we have the alveolar macrophages, and it removes the pollutants, microbes, and clears surfactants. That's a function it does. Then in kidney, we have the renal macrophages. They regulate inflammatory response to antigen filtered from blood. Okay. In brain, we have the microglia, and they basically help in the neural circle development for the synaptic pruning. That, that's a part of the nervous system. In liver, we have the Kupffer cells, and they scavenge red blood cells and clear particles. In spleen, we have the red pulp macrophages. They scavenge red blood cells and recycle iron. In the skin, we have the Langerhans cells, and they are important for the skin immunity and tolerance. In peritoneal cavity, we have the peritoneal cavity macrophages, which maintain IgA production by the B1 B cells. It's a kind of cells. Then in intestine, we have lamina propria macrophage and intestinal muscularis macrophage, and they are important for the gut immunity and tolerance, and they also regulate peristalsis, right? In bone marrow, we have the bone marrow macrophage. They maintain the niche for blood cell development, and they clear the neutrophils. In lymph nodes, we have the subcapsular sinus macrophages, which trap the antigen particles. And in heart, we have the cardiac macrophages, which clear the dying heart cells. Okay, so these are some of the tissue macrophages, the different types of tissue macrophages and their specific functions apart from, the regular function. Now, dendritic cells. Dendritic cells are covered with the long membranous extensions, and these resemble the dendrites of the nerve cells. So that's why their name is the dendritic cells. And these increase the surface area available for browsing the lymphocytes. So here you can see the dendritic cells, and on the surface you can see these extensions. These are just like the extensions which are found in the nerve cells like dendrites. But the beauty of these extensions is that they can be retracted and extended. That means they can go inside to some extent and then they can extend towards outside. So retraction and extension, these are the properties of these dendrites. Now the question is, why do we need this? We need this because these dendrites, they increase the surface area. And when these dendritic cells, because these are the antigen presenting cells, when they are presenting the antigen, so the antigen will be presented on these dendrites, and so a lot number of, T cells, they will have an opportunity to scan these dendrites and immediately, start interacting and activating. So that's why these dendritic cells are important. They arise from both the myeloid and lymphoid hematopoietic stem cell lineages. So we have seen in the initial slides that on both sides we saw the dendritic cell development. The distinct functions is to antigen capture and antigen presentation. So their main function is to first capture the antigen and then to present the antigen to the T cells. Outside lymph nodes, when they are not in the lymph nodes, and lymph nodes are the secondary tissues which we will see in the subsequent discussion. Outside lymph nodes, immature dendritic cells, they look for any pathogens to capture them. So they are the antigen capturing cells. So we call them as a ACC, antigen capturing cells. Their main role is to capture the antigen. So here you can see the antigen is being captured by the dendritic cells. Okay, so this is the antigen, red color, and it is being captured here. So they are the antigen capturing cells. Immature dendritic cells use three methods to take up antigen because they are immature, but still they can capture the antigens, and they use three methods. First is the phagocytosis. Then second is the endocytosis, where they internalize it by receptor mediated, endocytosis. And in case of the fluid, they imbibe it by pinocytosis. So these three methods are used to capture the antigen so that the antigen can be taken up and can be processed inside the cells, and then can be presented with the MHC class 2 molecules. So their maturation convert antigen capturing cells into antigen presenting cells. Now this is very important point, because by now the immature cells, they are the antigen capturing cells, but once they are mature, now they are the antigen presenting cells, and they have to present the antigen. So they lose the capacity for phagocytosis and pinocytosis, and they express co-stimulatory molecules which are important for activating T cells. Now the question is why it happens? Why suddenly the antigen capturing cells, they lose their ability of phagocytosis? Why? The reason is because they have the sample of the antigen, and they have to present this antigen to the T cells. If they will not lose this capacity, then probably it will keep capturing the antigen and keep processing it and keep presenting it with the MHC class 2 molecules. And in this capacity there might be a scenario where they get exhausted. Okay, and they probably would not be able to efficiently present a single antigen. So that's a reason probably the nature has downgraded the function of the phagocytosis and capturing, while they have changed these cells into the antigen presenting cells. Upon activation, the dendritic cells migrate from the tissues to lymphoid organs via blood or lymph to present the antigen to the T cells. So once they are activated, they migrate from the tissue, and they have to report this antigen to the T cells in the lymphoid organ. And this migration happens either via blood or via lymph, right? So here you can see the dendritic cells. This is the DC. It is presenting the antigen to the T cell. This is the T cell, right? We have the follicular dendritic cells, and these are different cells from the cells we just saw. These follicular dendritic cells do not arise in the bone marrow, first of all. They don't arise in the bone marrow, and they do not function as antigen presenting cells, or they don't take part in the Th cell activation. Okay, they are named for their exclusive location in the organized structure of the lymph nodes called lymph follicles. So when we will study the lymph nodes, we will see the structure of the lymph node, and in the lymph node there are organized structures, there are organized regions of the follicular dendritic cells, and these are known as the lymph follicles. And because they are present in the follicle, they are called as a follicular dendritic cells, and these cells are rich in the B cells. Okay, the lymph follicles are rich in B cells. They are critical for the maturation and diversification of the B cells. So their main role is to mature and diversify the B cells rather than presenting the antigen to the T cells. So that is the difference between the dendritic cells and the follicular dendritic cells. Then lymphoid derived dendritic cells are fully capable as a antigen presenting cells. So in the lymphoid side also, you saw that we have the myeloid branch, and then we have the lymphoid branch also, we have the dendritic cells. They are also fully capable of the antigen presenting cells. Activated B cells can also act as a professional antigen presenting cells. So activated B cells, they can also take up the antigen. They can also present it with class 2 MHC molecules on their surface, and uh, they can also activate the T helper cells. So they can act as a professional antigen presenting cells, and they also express all co-stimulatory molecules which are required to activate the T cells. So B cells, they not only can generate the antibodies by differentiating into the effector cells, but they can also become the antigen presenting cells. They can also engulf the antigen and present it to the T cells, just like any other and professional antigen presenting cells like macrophages or the dendritic cells. So here you can see the comparison between the macrophages and the dendritic cell. So both have the phagosomes, okay, and both have the processes, but here the processes are longer, okay, in case of dendritic cells, and here we call them as a pseudopodia because they are not that much longer. Here you have the phagosomes and lysosomes and phagolysosomes. Here you also have the phagosomes, lysosomes, and the phagolysosomes. The nucleus is very big or bigger in case of macrophages, while in case of the dendritic cells, the nucleus is smaller. So, these are some of the fundamental differences between the morphology of the dendritic cells and the macrophages. And both are the professional antigen presenting cells. Now come to the erythroid cells. Erythroid lineage form erythrocytes or the red blood cells. So here you can see the RBCs. They arise from the common myeloid progenitor, okay, or we also refer to them as common myeloid erythroid precursors because they are from the myeloid lineage, but they are committed to the production of the erythrocytes. So these are the common myeloid erythroid precursor. In mammals, they are anuclear. That means they lose their nucleus. So erythroblast is the precursor which extrude their nuclei in the bone marrow. Extrude means they simply squeeze out the nucleus into the bone marrow. So they become the anuclear cells, and that is the case in mammals. But in all non-mammalian vertebrates, for example in birds, fish, amphibians and reptiles, we have seen the nucleated RBCs. So their RBCs are the nucleated one. The largest RBCs are in some amphibians, while the smallest one in the deer species. So here you can see in case of frog, which is an amphibian, you can see here the RBC, which is the largest. Okay, but we don't have here for the deer, but you can see the comparable sizes. Damaged red blood cells. Now, it is important. Damaged red blood cells can also release signals, which are the free radicals that induce innate immunity. So, erythrocytes, their main function is to carry out the gaseous exchange and transport the gases, oxygen and carbon dioxide. But they can also take part in the innate immunity by releasing the signals like the free radicals. And then they also express surface receptors for antibody and bind antibody complexes cleared by erythrocytes scavenging macrophages. So what they do when they are in the in the blood, when the free antigen bind is opsonized by the antibody, then it is not only taken up by the macrophage, but these RBCs can also bind those complexes. And when they bind the complexes, then erythrocyte scavenging macrophages, they can clear the infection. So they also play a role in our immune response. Fine. Now, look at the functions of the megakaryocytes. These are the large myeloid cells. They reside in the bone marrow and they give rise to thousands of the platelets. Okay, so here you can see this is the megakaryocyte. It is giving rise to the platelets. And platelets are very small cells or cellular fragments that circulate in the blood and participate in the formation of the blood clot. So they are important in blood clotting. Platelets have some of the properties of the independent cells, but they do not have their own nuclei. Hmm? They do not have their own nuclei. Now let us focus on the cells from the common lymphoid progenitor, and the cells are the lymphocytes. They are the main cells of the adaptive immunity. So around 20 to 40% of the leukocytes or the WBCs, they are the lymphocytes, and the 99% of the cells in the lymph are the lymphocytes or the white blood cells, but are kind of white blood cells. There are three major types of lymphocytes. One are the B lymphocytes, then we have the T lymphocytes, and then we have the innate lymphoid cells, and one example is the NK cells or the natural killer cells. Their morphology is similar, so identified by surface proteins, which are the CD, cluster of differentiation. Now, what is this cluster of differentiation or designation? Now, you understand that we have like here I can draw, okay, we have an antigen, okay, and in antigen we have suppose three regions where the antibodies can bind. This is region 1, this is region 2, and here we have region 3. Three regions on this antigen where antibody can bind. Now what happens? We generate the antibodies, and three kind of antibodies are generated. One will bind here, one will bind here, and one will bind here. So we have single antigen, but we have three monoclonal antibodies maps. So I can write monoclonal antibodies. So these three antibodies, I will keep it in a cluster, and I will say this is the CD, and I will give this cluster as a number. For example, this is CD5. I'm just taking an example. So whenever I will refer to CD5, I am referring to the antigen which can be recognized by the cluster of these three monoclonal antibodies, and only these three monoclonal antibodies. Even if they are recognized by two monoclonal antibodies but not the third one, then this is not the right protein. So the condition is that the antigen should be recognized by these three monoclonal antibodies, and only these three monoclonal antibodies, and then this is known as a cluster of differentiation or designation, right? So every molecule or every antigen has a CD designation. We call CD1, CD2, CD3, CD4, CD5, CD6, and so on. In T cells, you must have remembered that we have the CD4 as a T helper cells and CD8 as a T cytotoxic cells. So that means the cluster of antibodies which belong to that particular CD, they all will bind to either that CD4 molecule or CD8 molecule, and no other monoclonal antibody should bind. Okay, so this CD designation is important, and it is defined as a cluster of monoclonal antibodies that appear to be binding to the same protein. Because protein is large, the antigen is large, so it has a number of sites where the monoclonal antibodies can bind. And all the monoclonal antibodies together, if we keep them in a purse, we call this purse as a, and we give the designation to this purse as a particular CD designation, like CD1, CD2, CD3. In the case of CD1, there might be 5 monoclonal antibodies in the purse. In case of CD10, there may be 100 of monoclonal antibodies in the purse. It all depends, what is the type of the protein it is, how large the protein it is, and how many antibody sites it has, right? So this CD designation or cluster is very convenient to use and to determine, and to see the cells and to select the cells and to isolate the cells. Besides these CDs, B and T cells express an antigenic specific surface receptor On B cells, we have the B cell receptor, and on T cell, we have the T cell receptors, apart from the CDs. Okay. B and T cells mature but unexposed to antigen, and such cells are known as the naive cells. So, whenever we say there are the naive B cells or the naive T cells, you should know that these cells have not been exposed to any antigen yet. Okay, that's why they are the naive cells. And these B cell receptor, it is the same antibody, which is released by the effector cells. So the antibody in the secretory form, when it is on the membrane, then it is known as the B cell receptor. And the T cell receptors are the unique receptors which we will come to know, and they also have a high level of diversity and specificity to antigens. So exposure induces naive lymphocytes to proliferate and differentiate into both effector cells, which carry out specific response, and memory cells. So whenever B and T lymphocytes, they get activated, okay, when they both get activated, they differentiate to form effector cells and memory cells. Effector cells function is to immediately neutralize the infection, neutralize infection, while memory cells, they are the long term cells which maintain the record of the infection, so that next time when the same infection will come inside the body, then these cells will immediately respond quickly and more effectively, more aggressively. You remember the primary response and the secondary response, and in secondary response, the memory cells are the main activators. Okay. So now just see what B lymphocytes are. So B lymphocyte development is not restricted to one particular organ. In human and mice, they develop in the bone marrow and mature in the spleen. In cattle and sheep, they develop in the bone marrow and mature in ileal pairs patch. In rabbit, appendix is the primary lymphoid tissue for their development. In jawless vertebrates, B lymphocytes like cells, not exactly the B lymphocytes, but B lymphocytes like cells, they developed in the distinct region which we call as the typhlosoles in the kidney and intestine. And in sharks, B cell development from liver to kidney to spleen. In amphibians and reptiles, liver and spleen to bone marrow. And in bony fish, it takes place primarily within the kidneys. So you can see, so B cell development is not just restricted to one particular organ. So in the animal kingdom, you can see so much of variation. Now coming to the T lymphocytes, why the name is T lymphocytes? Because their site of maturation is thymus. Okay, so here it is the thymus, you can see, and here it is, the thymus here. Now, this bursa fabricius is basically in birds the main organ of B cell or the B lymphocytes. B lymphocytes. So here is the thymus in case of humans. Mature T cells, they display TCRs or the T cell receptors, which can recognize processed antigens which are bound to major histocompatibility complex glycoprotein molecules. And we have seen two types. Class 1, as we discussed, these are expressed by all nucleated cells, and class 2, which are expressed by professional antigen presenting cells and some few other cells during inflammation. Okay, during inflammation. So these are non-professional antigen presenting cells. Two major T cell types, and both browse the antigen presenting cells. We have the T helper cells, which are the CD4 positive cells, and they recognize class 2 MHC bound antigen. And Once they recognize class 2 MHC bound antigen, they help in induction of the immune response by activating the T C cells and B cells. So here you can see these are the helper cells, CD4 positive cells. So whether we are saying the CD4 positive cells or we are saying the T helper cells, both are same. So here it is the TCR, and here the MSC class 2 molecule is presenting a peptide here. So TCR is binding with the class 2 peptide complex, okay, and then here this is the CD4 and it is recognizing. And in case of TC cells, which are also known as the CD8 positive cells or just CD8 cells, they recognize class 1 MHC bound antigen. And this class 1 MHC bound antigen, it is expressed by all nucleated cells, right? And here you can see this is the cytotoxic T cells. These are the CD8 cells, and this is the TCR, okay, and this is the MHC class 1 with the peptide, and this TCR is binding with MHC class 1 peptide complex. So the normal ratio of CD4 positive and CD8 positive cells is approximately 2 is to 1. So out of almost roughly 3 cells, 2 cells will be the helper cells and 1 cell will be the TC cell. This is the normal ratio. But whenever there is a change, then it also shows the clinical challenge or clinical issue. Now, one example is that in case of HIV, because HIV targets the T helper cells and it reduces the number of the T helper cells, so automatically the ratio disturbs and now the TC cell increases in the system, and that gives a picture that there is an infection, HIV infection. Okay. Now, MSC1 activated naive CD8 positive T cells, they proliferate and differentiate into the effector cytotoxic T lymphocytes or the CTLs. So the effector cells of the CD8 positive T cells or TC cells are the CTLs, cytotoxic T lymphocytes. So you should understand like this: TC cells, they get converted into CTL, and CTL is the effector cell. Okay, now CTL is a killing machine, it is the sniper. It kills any cell that displays foreign antigen complex with class 1 MHC molecule. Whether this is the virus infected cell or the tumor cell, they are killed immediately when they show the class 1 molecules with the antigenic peptide. So they don't think, okay, they are our encounter specialist. They simply kill it. Fine. Now, MHC2 activated naive CD4 T cells, because these are the T helper cells and they recognize MHC2 class of molecules, and when they get activated, they proliferate and differentiate into one of a variety of effector subsets. We have T helper type 1, so we call it as a Th1 cells. They regulate the immune response to intracellular pathogens. So Th1, they regulate intracellular pathogens. Then we have Th2 cells, they regulate the response to many extracellular pathogens. And two additional Th cell subsets have recently been identified. We have T helper type 17 cells, so we call it as a Th17 cells. Why? Because they secrete interleukin 17, and that's why this number is given to their name. We call it as a Th17. So remember, Th17 secretes interleukin 17 and it plays an important role in cell mediated immunity and may help the defense against fungi and intracellular pathogens. And then we have the T follicular helper cells, TFSH. It plays a role in the humoral immunity and regulate the B cell development in germinal centers. So you remember the germinal centers are okay. So we have not discussed that, but you remember the follicular regions. These follicular regions, when they have the activated B cells, they simply differentiate into some germinal centers, which are the site of the interactions, or the B cell maturation and their immune responses. So these germinal centers, they are generated and they also respond to extracellular pathogens. Okay. So we have these four T subsets: T helper type 1, type 2, T17, and then T effector. Then we have another type which is the T regulatory cell. They have the ability to inhibit immune response, because by now you are seeing that every time whenever we are talking about the cell, we are only talking about the induction or the stimulation of the cells and the immune response initiation. But once the pathogen is killed, then we need to stop and we need to inhibit this response. And for this inhibition, we have this T regulatory cells. So that's why we call it as a Tregs. And now, over the period, these Tregs are really, coming up with a quite interesting factors, and they have the ability to inhibit the immune responses. There are many other subsets, they have been identified. So if you find the list, we have a number of lists, and the list is continuing and increasing, because we are now coming up with new subsets. But that's not the scope of this lecture. We can just give you a glimpse here that the T helper cells, they have a number of effector cells, and these number of effector cells, they do a different function. So you can see here, T helper cells are also regulating the intracellular pathogens. They are also regulating the extracellular pathogens. They are also mediating the cell mediated immunity. They are also controlling the fungi. They are also mediating the humoral immunity. So they are also regulating the B cell development. So basically you can see they are helping, and that's why the name is the helper cells. Okay, the name is pretty good, and that suits to these cells. So let us, focus 2 points on the regulatory T cells or Tregs, which have the capacity to inhibit an immune response. We have the natural Treg, which arise during maturation in the thymus from autoreactive cells. Now you remember that in our first class we spoke about the tolerance, okay, where the autoreactive cells were deleted from the system. So how they were deleted? Because these autoreactive cells, they were deleted with the help of these Tregs, and they come up in the they mature in the thymus from the autoreactive cells, because they know where to stop. And then we have the induced Tregs. These cells, they induced at the site of an immune response in an antigen dependent manner. So basically these are the Tregs. They are quiet, but when they are at the site where the immune response is there, then in the antigen dependent manner they are induced. Okay. The Treg markers are CD4 positive, which are the surface markers, CD25 positive, which is again a surface marker, and then internal transcription factor, we have FOXP3. So FOXP3 positive, which are internal, CD25 and CD4 positive are the external surface markers of the Tregs. So if we utilize the monoclonal antibodies against CD4, CD25 and FOXP3, then definitely we will be able to isolate these cells. These suppress autoreactive responses, and they may play a role in limiting normal T cell responses to a pathogen. So they are pretty important for our immune response, because they are limiting the normal T cell response to a pathogens. Otherwise, T cell will keep responding, and it may cause the immune dysfunction. So they control the immune dysfunction, and they optimize the immune response. So which helper subtype dominates a response? It depends largely on the type of the pathogen, what kind of pathogen it is, and what kind of our T helper subtype will come. If it is an extracellular pathogen, then we have two sets. If they are the intracellular pathogens, then we have two sets. If we have fungi, then we have the set. So depending on what kind of pathogen is there, the T helper cells, they form the effector cells. Each subtype produces a different set of cytokines that enable the activation of B cells, TC cells, macrophages, and various other cells that participate in the immune response. Because the main function of the T helper cells is to help to induce the response. Now, what kind of response to be induced, that depends, what kind of pathogen it is. And once the pathogen decides the response, then accordingly they induce the B cells or T cells or macrophages and various other cells in combination or independently, depending on what kind of situation is. Now we have an interesting natural killer T cells, or we call them as a NKT cells. They have the features of both the innate and adaptive immunity. And like T cells, these NKT cells have T cell receptors. Okay, we have you have seen the T cell receptors which interact with MHC molecules. Okay, type 1 and type 2, and these NKT features, some express the CD4. So CD4, again, it's a marker of the T helper cells, right? So they have the TCRs as well as some, not all. Remember, they express CD4. But unlike most T cells, unlike, not like most T cells, the TCRs are not diverse. In case of TCRs on the T cells, they show enormous diversity. They can bind to almost any kind of antigen peptide, but they have very limited diversity, and they recognize specific lipids and glycolipids. While in case of T cells, they were recognizing the peptides. Remember, the part of the protein. But here they are recognizing the lipids and the glycolipids. And these are presented by CD1, which is a relative to MHC molecule. While in case of T cells, the peptides were presented by the MHC molecules class 1 and class 2. So that is the major difference between the NKT cells, and it has both innate and adaptive immunity. So their TCRs are of limited diversity. First point. Second point, they recognize lipid and glycolipids present on the CD1, and so they are presented by CD1, not by MHC molecules here. They also have the antibody receptors as well as other receptors classically associated with NK cells or the natural killer cells. So now these 2 points, the this one point, it is giving a T cell like feature, and this is giving the natural killer cell like feature. So if we combine these, we make the NKT cell, natural killer T cells. So they have the antibody receptors as well as other receptors which are classically associated with the natural killer cells. Activated natural killer cells can release cytotoxic granules that kill target cells, but they can also release large quantities of cytokines that can both enhance and suppress the immune response. So they are very, very critical cells. They can release the cytotoxic granules. They can degranulate and kill the target cells, and they can also release the cytokines which can increase or inhibit the immune response. They appear to be involved in human asthma. Human asthma causes by degranulation of histamine, because histamine constricts the bronchial passage, and it causes the asthma. So they degranulate, but they may also inhibit the development of autoimmunity and cancer. So on one side, their degranulation is causing a kind of disease here, or a kind of a response, I would say, maybe because of some transient response due to some dust or some pollens. So it is giving asthma kind of symptoms, but they may also inhibit the development of the autoimmunity and cancer. The T cell receptor on many human natural killer T cells is invariant. They don't vary much. And with the TCR alpha and TCR beta chains encoded by specific gene segments. Now, this is something why we have the T cell receptor and B cell receptor as a diverse because we have different gene rearrangements. We will see in the later classes where we will discuss the details. But here you need to understand that the T cell receptors on the NKT cells, it is invariant. That means it doesn't change much because it is decoded by only specific gene segments. So all the NKT cells will have almost the similar type of the T cell receptor, not diverse. And these cells are referred to as the invariant NKT cells or iNKT cells. Okay, so iNKT cells are basically a subtype of NKT cells which shows very limited diversity or very limited variance or almost no variance in the T cell receptor. So that's why they are known as the invariant NKT cells. Now we have the innate lymphoid cells, and these are a group of cells that are derived from common lymphoid progenitor but do not express antigen specific receptors. So that's why these are the innate lymphoid cells. Although they are originating from the lymphoid progeny, but they are not expressing the antigen specific receptors in case of the like in case of T lymphocytes and B lymphocytes. We have three groups, ILC1, ILC2 and ILC3, on the basis of the cytokines they secrete. So these groups secrete some specific cytokines, and based on these cytokines we have grouped them into ILC1, ILC2 and ILC3. So here you can see group 1 ILC, its major part is the NK cells and ILC1 cells, and here the signature cytokines secreted by CTL, which are the T cell subsets, because they are the lymphoid cells. So they have the IFN gamma, perforin and granzyme, and ILC1 has the TH1 cells which release the IFN gamma and TNF. In ILC2 cells, which having some relativity with Th2 cells here, IL4, IL5, IL13 and amphiregulin, and in the type 3 or group 3 cells we have IL17A, 22 and LT alpha and beta, and in this ILC3 we have IL22 and IFN gamma. So these are the T cell subsets which are under consideration in different groups, and these are the signature cytokines which are secreted by both the T cell subset and the specific ILC. So natural killer cells are the best studied cells, and all the natural killer cells, they express NK1.1 receptor. This is the signature receptor, or this is something the specific natural killer cells specific receptor, NK1.1. So if you see the natural killer cells, they kill the MHC1 deficient abnormal cells. All the nucleated cells, they have the MHC1 expression, and in normal condition all the cells display some MHC1 molecules, some, not higher expression, but a very lower housekeeping level of expression. And these cells, they keep browsing, all the cells, natural killer cells. And they, whenever there is, the MHC1 expression, the MHC1 molecules, they interact with the receptor on the natural killer cells. Okay, and they leave the cell. But when there is some problem in the cell, the MHC1 expression goes down below the threshold. Okay, like in case of the virus infected cell or in case of the tumors or cancers. So in those cells, when MHC1 expression is down, the natural killer cells can easily recognize them and immediately kill them because they don't find the minimum threshold level of MHC class 1 expression on the cells. So any cell which is deficient of MHC class 1, it will be killed by the natural killer cells. So they express the Fc receptors for some antibodies and bind antibodies and find target cells. Now, this is also the beauty. They have the Fc receptors. Suppose here we have the Fc receptors. Okay, this is Fc receptor, and they bind antibodies. Now, since this is the Fc receptor, they will bind an antibody. Okay, we have bound here the antibody. Now, these cells will go and they will look for the antigen for this antibody. And whenever they will find the antigen will be bound here, and it will cause some conformational changes. And then they will degranulate the content, because natural killer cells, they have the granules. They will degranulate and kill the cells, and this is known as antibody dependent cell cytotoxicity, or in short it is known as ADCC, antibody dependent cell cytotoxicity. So these are the functions of the natural killer cells. Okay. So these natural killer cells are different from the NKT cells, because NKT cells are kind of hybrid cells bit of the cells which have the properties of both the T cells and the natural killer cells. But these are the real natural killer cells. So I think we are closing the class today, and let us summarize what we have learned so far. So we covered the hematopoietic stem cells. We covered hematopoiesis and its regulation. We talked about the transcription factors, how they regulate the hematopoiesis, and how hematopoietic stem cells, they further, progressively commit themselves into specific lineage. And we also, saw the myeloid and the lymphoid lineages and the cells, their properties and their role in immunities. So basically we covered the immune cells, their characteristics and their functions, because in our future discussions these will be of our use, and we will keep discussing, and we will keep coming up with the functions they will be producing when we will study the immune responses in the future classes. All right, so I hope that you have got some information about the immune cells, about their roles and their functions, right? In case if you have some feedback, you can always reach out to me, and we will meet in the next class. By the time, take care and thank you.